Before we begin, I would like to start by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. So welcome everyone, my name is Anya and I will be chairing the seminar today. I'm a PhD student of the CCMP Center at the Monash Node. And before we start and introduce our speaker, I would like to quickly go over some housekeeping rules. So first and foremost, I'd like to let you know that today's seminar will be recorded and you will be able to find this as well as previous recordings on the CCMP website. The presentation by our speaker today will be 45 minutes, followed by 10 to 15 minutes Q&A session. So please leave your questions till the end of the seminar, either by raising your hand or typing them in the chat box so we can address them later. Please also make sure your microphone is muted throughout the presentation. So our speaker today is the CEO of Structura Biotechnology from Toronto, Ali Punjani. Ali has background in engineering and he completed his master's at the University of California, Berkeley in computer science and machine learning and subsequently pursued his PhD at the University of Toronto in computer science, where he worked on novel cryo-EM reconstitution algorithms. The development of these methods formed the basis for early versions of CryoSPARC, and research work has been ongoing ever since at Structura Biotechnology. So the talk today will be about methods and tools in CryoSPARC for processing of membrane protein cryo-EM data. So over to you, Ali. We are very lucky to have you here today, and the screen is all yours.
Amazing. Thank you so much. And before I start, I just want to give a huge thanks to the organizers and our chair today for the very kind introduction and the invitation to speak. It's really an honor, and I'm very excited to talk to you today about new developments in CryoSPARC. CryoSPARC is a software system that's used for processing single particle cryo-EM data. It's quite widely used at this point, and specifically today, I'll touch on some new developments and tools that may be interesting and useful for those working on membrane protein cryo-EM structures, and hopefully that'll be relevant to the audience today. Awesome. So before I start, I just want to say that everything I'm presenting today is on behalf of the entire team at Structura Bio, and that's everyone you see here. And these are really the people who develop, distribute, and maintain CryoSPARC, and all the credit for the new developments, implementations, and experiments that we have that we're showing today goes to them. I also want to say a huge thank you to any CryoSPARC users that are out here in the audience today. It's really your support, feedback, and the success stories that we hear from you that keep us motivated to continue developing new methods, new tools, and software. We've been working on CryoSPARC really since before we released the first version back in 2017. There's been many new versions, new features, interfaces, software systems, new methods, etc., that we've added to CryoSPARC over the years. And of course, over that time, single particle cryo-EM itself has gone through a lot of change and development across the board in terms of methodology, in terms of cameras, detectors, microscopes, sample preparation, and everything that's part of the whole process. And from the start, really, the application of cryo-EM to membrane protein structure determination has been a huge focus for the field. And I think it's fair to say that probably everyone in the audience will know already about the strong upward trajectory and progress of single particle cryo-EM. The technique is really maturing quickly, and the diversity of use cases that we're now seeing around the world and across biological research is really growing very rapidly. The graph on the right, which again, many people will be familiar with, shows the number of new EMDB entries released per year over the last many years, and you can see that that trend is nothing but impressive. So what we're seeing, and what we often think about as sort of the next future, or one of the potential futures of single particle cryo-EM, is that the technique will become a ubiquitous technique that forms the inner loop of when researchers start to ask larger biological questions. Single particle EM becomes the inner loop of that process. So some examples of that, a context where single particle reconstruction is often repetitively applied are cases like synthetic biology, time-resolved cryo-EM when we want to solve multiple structures of perhaps the same target, but at different time points. And of course, the quintessential example, which is drug discovery, trying to solve many structures with different ligands bound. In many of these cases, it's obviously important to note that when we're starting an investigation in any of these scenarios, we often know a whole lot about the target molecule that we're looking at. Unlike the early days of cryo-EM, when often many of the discoveries were simply to solve a first structure of a new target that had never been visualized before, nowadays there are many very interesting and important biological questions that can be answered with some prior knowledge of the structure. This is also equally important to keep in mind that developments in other computational areas of structural biology, especially structure prediction, have made it such, with AlphaFold, for example, have made it such that we often do have some initial information when we're starting a cryo-EM project. In many of these cases, of course, membrane proteins are the targets that we're talking about, given their relative importance for things like signaling and other cellular activity. In all cases, rapid automated structure determination is something that we foresee and many foresee is likely to be an important accelerant for new discoveries. And in fact, this concept has been important to us in the development of CryoSPARC really right from the start, both in terms of speed and simplicity of the software and the user interface, of course, but primarily in terms of methods development. Within CryoSPARC, we spent a lot of time over the years trying to develop automatic parameter tuning methods, schedules for optimization techniques, diagnostics, etc., so that users generally need to provide as little input as possible to get a reasonable structure out of their data. And of course, there are still many manual steps in processing of cryo-EM data for state-of-the-art results. Recently, we've been working on taking this picture a little bit further forward towards the goal of automated single particle analysis. And that's actually what I'll talk about today, specifically with a focus on membrane proteins. Along the way, I'll also highlight a few new developments in CryoSPARC, some of which were recently released and some of which have not yet been released, but we hope to release soon. As a demonstration and example of moving towards further automation, I'll focus on active GPCRs as a target class. This is obviously a very important class of targets, especially for the repeat structure determination scenario where there are multiple different ligands being tested, and of course, particularly for drug discovery, since already something like more than a third of all approved drugs target GPCRs. These are typically relatively challenging targets for cryo-EM due to their small size and other various problems, such as inherent flexibility, the often presence of lots of junk particles in a data set, denatured particles, broken particles, empty micelles, etc. I'll be walking through the processing and demonstrating some automated processing results on a set of public Empire datasets that all have some different characteristics, and I'll walk through all of those in a minute. I just want to outline the sort of scope of what we're talking about here. Of course, automation of single particle cryo-EM data processing is not a new concept. Many people have been thinking about it and trying various ways to approach that future possibility in various ways. And typically that's been done in a sort of generic manner before with the development of, for example, methods to automatically pick 2D classes, or for example, a method that may be a generic particle picker that doesn't require any training, but can produce reasonably good particle picks on a new data set that has not been seen before. All of these techniques have been sort of in the bucket of having a goal that we're trying to produce a first cut structure that can then be improved by manual processing. The goal that we have here today and what I'm going to show is a little bit different. We don't just want to get a first cut structure. Ideally, we want to see if it's possible to get close to the level of quality, map quality, and resolution that can be achieved by manual iterative processing, but completely hands-off. And it's important to note that this is required really in the context of the larger biological problem-solving situations where we really want single particle results to be available immediately and ready to use as part of that inner loop of a larger question. So how can we go about this kind of a question? Our approach is relatively simple. We're going to make use of some limited but very important prior structural knowledge about our class of targets. So for the case of active GPCRs, for example, we will make use of a generic coarse resolution reference structure. And this is really a 15 angstrom, very low resolution, what you see in gray here, coarse reference structure that is a sort of generic template of a GPCR, an active GPCR, as well as some sort of decoy densities. And we'll come back to what that means and how those are generated that are fixed and used across the different data sets that we're going to be talking about. None of this prior information that we have is really specific to a particular GPCR dataset. Rather, it's general prior information that we have about this class of targets. This, of course, makes sense for GPCRs since as a class, they typically have a lot of structural similarity. And you can imagine how this concept might be applicable for other classes as well, perhaps ion channels or spike proteins of a certain virus, etc. So first, I'll just walk through the 8 different data sets that we will be processing and looking at today. These are all public Empire datasets, and actually a couple of them were collected in Australia, so they may be familiar to audience members. They range in their nominal resolution. These black numbers are the deposited EMDB resolutions that were deposited with the structures that were solved. And they range from 2.1 to 3.5 angstroms in nominal resolution. The sizes of the datasets are also quite variable, ranging from around 2,000 to around 12,000 movies, depending on the dataset in particular. What's also interesting to note, if we just flip back a slide, the color scheme that's in common used here, fabs are highlighted in orange. Each structure actually has a fab, and they are all in slightly different positions and obviously different fabs. So the structures are quite highly related, but they do have a relatively sizable chunk of density that's different in each case, which is the fab. And of course, the extracellular domain is quite different, and the transmembrane domain structure will be different. 7 out of 8 of these have a ligand present. And so they have quite a bit of variability. You can already see just in the renderings of the EMDB structures that are shown here, the deposited structures from the original authors, that there's quite a variability in the quality of the maps as well, as that corresponds with the resolution. In some cases, you can see some effects of preferred orientation. In other cases, perhaps quite high quality density in the transmembrane domain, etc. So this is what we're starting from. The first new development in CryoSPARC that really allows for automation and automated processing that I'm going to talk about is a new software system and a new user interface system that's called Workflows, aptly named. This was released in CryoSPARC version 4.4, and a workflow you can think of as an extension to the CryoSPARC job builder for those who've used CryoSPARC before. What it allows for is selecting an entire chain or tree of processing that you've done before or have just set up before, saving that with various annotations, default parameters, flagging parameters, etc., and then being able to reuse an entire chain of processing in a single click. The purpose is partially for automation and time saving for those who are doing repeat processing, but also for ensuring things like repeatability between researchers, and especially for training. We see already that expert users are often creating workflows that are then shared amongst a lab with newer users who are just learning cryo-EM data processing, so they can get a sense for what an expert user might do with a particular data set. Workflows can be easily exported and shared from one CryoSPARC instance to another, and can be easily reused within a CryoSPARC instance across workspaces and projects. I'll just give a little quick demo in this video here showing how a workflow works. So this is an example where several jobs are selected at once. So you can, with one click, select all of the jobs that were spawned from this select 2D. And these jobs can be all linked together into a workflow. This workflow is now titled Refinement Fork here. You can give it a category, add labels, annotations, descriptions, default parameters, etc. So this workflow is now created, and you can see that it appears on the right-hand side. The next video shows how this workflow can be, sorry, I'll go forward. This video now shows how this workflow can be applied in a new context where you want to do the exact same processing again with different data. So we can simply select the job to start from, find the applicable workflow. You'll notice that the selected job is highlighted in green, set any parameters and decide whether or not to queue the jobs immediately or queue them later, set any parameters that are different from defaults, and in a single click, apply the workflow, which will then create and connect all the jobs that are needed for processing. You'll see that happen in just a moment. There we go. So workflows are a very versatile and powerful tool, and we're going to use these to start thinking about automation in the context of our GPCR datasets. Now, for our GPCR datasets, the workflow overall of processing is going to be relatively straightforward for those who've done cryo-EM processing before, and consists of 3 basic phases: pre-processing, including particle picking, and then particle curation, which for challenging datasets can often be the most difficult part, where we need to separate high-quality particles from less good particles during our particle picking and curation, for example, a 2D classification and so on, and try to separate out a subset of homogeneous particles that can refine to high resolution. The final stages will be refinements to hopefully achieve those high-resolution structures. So within pre-processing, our workflow can begin with the relatively standard steps of patch motion correction and patch CTF estimation in CryoSPARC. These are tools that are very widely used and are fully automatic. They don't have any parameters that need to be tuned on a per dataset basis, and they correct for both beam-induced motion, stage drift, and estimating the CTF of samples that may not be perfectly flat. For example, if you have images at the edges of holes, which are where the ice is often curved, or some residual tilt in your stage, or if your grid is crinkled, etc. Patch CTF and patch motion will take care of all of those problems. Even so, once we do that initial preprocessing, we can see that our data sets are very large, and some micrographs are just going to be poor right off the bat. And those can be excluded with simple thresholds on things like CTF resolution, total motion estimates, using the typical exposure curation pipeline in CryoSPARC. Still, at the end of all of that, we're going to have images that may contain some good particles, but also contain junk in the form of aggregation or ice buildup, carbon edges, etc. And in this scenario, especially with GPCRs, where there are often many broken particles, empty micelles, and other objects that we don't want to pick, particle picking can still be a challenge. And this brings us to the next new development that was just very recently released in CryoSPARC, version 4.5, which is a new micrograph denoiser. There have been many projects in the cryo-EM field working on micrograph denoising methods, and nearly all of them have been based on what's known as the noise-to-noise concept. The noise-to-noise concept involves taking raw data, for example, a cryo-EM movie, and splitting it into two parts. So the typical way of doing that is to separate all of the even numbered frames and the odd numbered frames in our movie data to produce two separate movies, each with half the number of frames, and then process each one of those separately to arrive at two different micrographs that are actually images of the same field of view, but with different noise. Once we have that set up, the noise-to-noise framework allows us to train denoisers and other useful tools that compare those two examples of noisy images that contain the same signal. Within this concept are tools, for example, in warp and topaz denoise and other denoisers that have been developed over the years. We've developed a new denoising method that's also based on noise-to-noise, but it has a couple of useful, interesting characteristics that make it practically really powerful, we think. The first is that micrographs are pre-corrected for CTF, and this means that the denoiser output will look similar whether your input is a low defocus or high defocus micrograph. This makes it much easier to inspect particles across a wide defocus range. We also found that during the training of the denoiser, combining this noise-to-noise denoising scheme with a classic computer vision technique called total variation regularization, that's sort of a classic, somewhat old school denoising technique. Combining these two, and I won't go through the math of what total variation means, but combining these two produces high quality denoised images and speeds up training and convergence of the neural network that produces the denoised images. Total variation regularization essentially is a technique that applies a penalty to the denoising training that attempts to encourage the denoiser to produce images that are as smooth as possible, except for the specific locations where they need to have quick jumps in their image contrast. So this would be around the edges of particles and where there's strong contrast. This helps to keep backgrounds very smooth and particles really sharp. I'll show some examples of that in a moment. The new denoiser that we have in CryoSPARC is very fast to train. It's fully unsupervised, so there's no manual labeling. You can just use raw data directly to train it. It takes only about 3 minutes on a single GPU to train, and it's quite data efficient as well. It requires only about 100 micrographs to train on a new data set. The great things about the denoiser output, and I'll show what that looks like in a minute, is that it actually tends to learn repeated particle structure, flattening out regions of an image that don't correspond to particles and accentuating the contrast of the repeated structure that typically are particles. This means that simple particle pickers, such as blob picking and template picking in CryoSPARC, work really well on denoised micrographs compared to running on raw micrographs. And one of the notable points of that, for those, again, who've done some processing of cryo-EM data, when you look at the particle picking statistics in CryoSPARC, if you've done, let's say, template picking on denoised micrographs, you'll often see a pattern that looks like this, where a cloud of high-quality particles is very clearly separated from a cloud of incorrect particle picks, and those can be separated really easily based on the typical normalized cross-correlation and power score that we use in the inspect particle picks procedure. So I'll now show just some examples of the denoising. In each case, I'm showing a triplet of images. The first is a raw micrograph. The second is a filtered micrograph. This is using the typical Fourier space filtering that would have been applied if you were to just visualize this micrograph in CryoSPARC normally. So this is kind of the best we can do just with standard signal processing techniques. And the third image is the denoise image. That's the output of our new micrograph denoiser. So this is one example. I'll just flip back between the filtered and denoised images so that you can see the difference. What you see here that's quite clear is that the background is heavily filtered, and a lot of the noise that otherwise impedes visual clarity for this micrograph is erased. But the particle objects and things that are or aren't particles, we don't know yet in this data set, are accentuated and maintained in the output image. I'll show another few examples. This one is actually a GPCR. So this is a raw image, a filtered image, and finally the denoised image. So again, you can see that the particles themselves are, their contrast is maintained quite strongly. In some cases, you may even recognize some views of a particle that you're looking for, versus the filtered images where that visual identification is quite difficult. A few more examples: raw, filtered, denoised, raw, filtered, denoised. This one is a particularly interesting example. I believe it's aldolase, quite a crowded micrograph, but the denoiser, if we look at the filtered image versus the denoised image, the denoiser is able to pick up the repeating pattern of the actual particles and the different shapes that they take on in terms of 2D projections, and accentuate those while flattening out the background and empty areas that don't actually contain any particles. Finally, some helical structures, raw, filtered, and denoised. Quite a stark contrast there. And raw, filtered, and denoised. So in these examples, we can see that particles are now showing up much more clearly, and hopefully particle picking becomes easier as well. But there are still other things in our images that are not helpful. For example, in this case, a really strong carbon edge. There are often examples of transfer ice and ethane contamination and so on. And this can still be challenging, especially for automated processing where we don't have a person looking at the micrographs during the processing. And this brings us to our next new development in CryoSPARC, which is something that is not yet released, but we hope to release a version of this soon. This is a new development that is particularly helpful for unattended processing, as I mentioned. And this is a micrograph annotation tool, also known as a junk detector. What we did here is to produce a neural network-based, machine learning-based semantic segmentation technique that looks at a micrograph and tries to label the objects in it belonging to a few different categories. So this is not a junk detector in the sense that it simply labels a micrograph as being good or bad. It will take any micrograph and label the specific parts of the micrograph that have that contain a certain object. And in doing this, we decided on 4 different categories of junk that we want our junk detector to be able to understand. I'll just explain what those categories are. And on the right, you see some examples of manually labeled images that we produced. We produced a data set internally of micrographs, mostly public data, that were labeled by our team and used that to train our junk detector. So we used 4 different categories. The first one is carbon and gold. That's a relatively self-explanatory category, simply edges of carbon and gold holes. Sometimes if there's lacy carbon or other things, they may not be really circular, they may be other shapes. Then there is the intrinsic intrinsic issues within the sample. This contains things like cracks, for example, highlighted here in blue, devitrified ice, wrinkles in, for example, a graphene layer, anything that is an aberration in our sample that's intrinsic to the sample itself. Then we have extrinsic issues. So this would contain things like ethane contamination, transfer ice, hexagonal ice. These are aberrations or objects that are on top or below our sample that are producing contrast in the image that's not helpful. And finally, we have a category called non-physical. These are aberrations or parts of an image that we don't want to look at because they contain, for example, beam fringes, gold reflections, residual stage drift, etc. And this is actually a relatively rare category. So we created a junk detector that's based on, we tried a few different neural network architectures, and I'll touch on that in a second. But we trained it from scratch. And one interesting thing that we found when doing this is that it's actually a lot more difficult than it seems to successfully label carbon. And the reason for this is that carbon, especially thin carbon that is used typically in grids today, looks a lot like thick ice if you don't see the edge of the carbon. So if you're looking at a part of an image that is simply thin carbon with particles on top, that often looks like very thick ice with particles in it. And it's actually quite difficult to disambiguate those unless you can see the edge of the carbon. So what we found when we were training this junk detector is that certain neural network architectures, for example, UNets, which are typically used for a task like this, actually struggle a lot with the carbon problem because they have an issue of forgetting the nearby context of a region of an image that's being classified. Instead, we chose to work with a newer architecture for these kinds of segmentation networks, known as a vision transformer. I won't go into the details of that, but it effectively allows for much longer range dependencies to be captured by the neural network and remembered, so that the fact that there's a carbon edge on one side of the image helps the network to remember that there must be carbon on the other side of the image. So once we train this junk detector, and I'll show some example results in a moment, it works quite well. Inference is very fast. It only takes less than half a second per micrograph. And it's great because it provides immediately with new data, several data set statistics, like what fraction of our images have cracks, what fraction of our images have ethane contamination, etc. And more importantly, it can allow us to automatically reject incorrect particle picks that are nearby to junk or on top of junk and mask away that junk during pre-processing as well. So I'm going to show just a couple of examples. These are 4 micrographs from 2 different data sets, just showing the raw filtered micrographs here. And these are the junk detector results. So in each case, you can see that the junk detector successfully labels, for example, the carbon edge and carbon region, large ice crystals, ethane contamination, transfer ice in various cases, cracks are labeled well, and large chunks of ethane contamination, etc. These images, by the way, are all part of a test set that was not used during training time. Okay, so with this junk detector in place, we can now try it on our 8 different data sets that we're working on today. And we can see that in all the cases, it actually works quite well. It's able to label, these again are not part of the training set. It's able to label, I think, contamination in all the different categories that we talked about. And this helps a lot to immediately remove particle picks and any objects that are not our protein, clearly. And so that our particle picking can be focused only on the actual protein objects, or perhaps not protein, or perhaps not our target protein if there are some denatured or broken particles in there. And that remains the next challenge. So once we have our 2 pre-processing new pieces, denoising and junk detection, we can then go on to a very simple procedure for picking particles. The strategy that we use is, and this is the first place really where our prior knowledge about the protein that we're looking for comes in. We need a diameter of our particles, which of course is again quite similar across the GPCR, active GPCR class. We use blob-based particle picking, just knowing the particle diameter. And then we extract particles from a subset of the data. I think in this case, we used 350 micrographs in each case. We perform 2D classification. And then we have a new tool, again, released in CryoSPARC version 4.5, which is called reference-based auto-select 2D. This is a very simple tool that takes our reference structure. This is our 15 angstrom, very coarse generic reference structure of a GPCR, and compares it to each of the 2D classes in our 2D classification result that came from automatic blob picking, and simply selects those classes that match the shape of our reference structure. The version of reference-based auto-select 2D that we used in these results is a slightly improved version compared to what's released in CryoSPARC right now, but we plan to release this soon as well. So using that generic 3D reference structure, we use a shape-based matching score simply to capture the correspondence between 2D classes and align projections of the 3D reference. And then those classes that match well are selected, and the ones that don't match are rejected. But what's important here is that we don't actually use the selection of these templates or classes for anything other than using these selected templates to then go back and do template-based particle picking on the denoise micrographs. So we don't actually use the selection of particles that went into this selection. We only use the selected templates to go back and do template-based particle picking. And this is an important point because we found, and many others in the community have found as well, that performing 2D class selection to separate good particles from bad particles can work well, mostly for larger and soluble proteins. But when we're looking at smaller proteins and especially smaller membrane proteins, where certain views are rare and certain views end up looking simply like circles, like with a GPCR. A top view, mostly you're just seeing the micelle. In those cases, being too aggressive in throwing away what appear to be junk particles at the 2D stage can often lead to orientation bias, since we're only selecting views that are very clearly for target structure, and we end up missing things that are actually our protein, but we just don't recognize them at the 2D stage. So we prefer actually to do the curation of particles all at the 3D stage. And again, this has been found by several members of the community and ourselves in testing that for smaller particles, removing junk particles in the 3D stage is quite a bit better. So our particle picking, just to recap, starts with blob-based particle picking using the particle diameter, reference-based selection to select templates from 2D classes using those blob picks, and then using those selected templates to redo template-based picking on the denoise micrographs. And after that, all remaining particle curation is done in 3D. So I'm just going to show some results of what this looks like on these 8 different data sets. And again, these are results that are automatic in the sense that we didn't do anything manual here. We just use our single shared reference structure across all the data sets and the same particle diameters across all the data sets. So for each one, you see the examples of classes that were selected automatically, and then the 2 numbers below, which shows you the number of particle picks after template picking across the whole data set. And then the second number is the number of particle picks that remain after filtering out all the junk. So empty ice picks from the denoiser, anything that hit our junk mask from the junk detector, and any particles that are too close to the edge. You can see that on some datasets, much more junk is removed than others. And likewise, on some datasets, the number of particles can still be very large, in the millions at this point. Okay, so at this point, our particle picks are good, but not great. We still have many poor particles in the mix, particles that may more or less look like our target structure, but may be broken, denatured, or otherwise not useful in some way. And here we use another strategy that we and many others in the community have found to be effective. That is known by 2 different names, decoy classification or baited classification. The idea here is that we can classify our particles into a few different classes where the initial structures for those classes are provided by us specifically for the task of separating good particles from bad ones. So what we do is provide 5 different starting volumes, and those are shown here in the top row. The same 5 volumes are used across all the data sets. One of them is our correct intact reference structure of a GPCR. The other 4 are what we call decoy classes, and they're designed to attract particles that are not quite correct according to our reference. So the first 2 decoy classes we create by hand, and those are simply using the reference structure and erasing different parts. So the first one is erasing everything except the micelle. The second one is erasing the entire transmembrane domain and leaving only the G protein. The last 2 decoy classes that you see on the right are randomly generated noise, effectively. But we randomly generate these once, and now all these 5 starting structures, these decoy classes, are fixed and used across all the different data sets. So in our first step of cleaning up our particles, we use these known starting classes and 1 round of heterogeneous refinement, using 1 good class and multiple poor initial volumes. What this yields is a classification and alignment of all of our particles, often in the millions, to these different classes. And the particles that match our reference structure, our correct reference structure best, are the ones that we take forward to the rest of the processing. The particles that best match our other classes, we discard at this point. Once we have the first cleaned up set of particles, we then go on to another round of 3D cleaning of our particles. And here we use a different strategy where we simply take all of the particles that are in this good class from the heterogeneous refinement and apply multi-class ab initio reconstruction in CryoSPARC. So here we are no longer using a reference structure. We're starting from scratch again, throwing away all of the particle poses, simply taking this set of particles and producing 5 ab initio structures. What happens here is that the particles, again, classify and align into clusters, each one being one of these classes, and typically 1 or more of those structures will be our intact GPCR protein. But this structure is formed ab initio, and so it may not be aligned to our reference structure, and we don't know which of the 5 classes actually contains our intact structure. So once we're done ab initio reconstruction, some of the classes, again, will be junk particles or structures that are meaningless, and some of them will be our intact structure. In this case, there's 1. So we take all of those and connect them again to another round of heterogeneous refinement. This time, we're simply refining the classification and structures as we go. And again, all of the good particles we hope will aggregate into 1 or more classes that contain our object that we're looking for, in this case, the GPCR, and the junk particles will stay in these other classes that are left over. After this point, we can apply another new tool that was released in CryoSPARC version 4.5, reference-based auto-select 3D. And here we can go back to our reference structure and use reference-based auto-select 3D to compare it against all the 5 structures that came out at the end of this cleaning pipeline and select the 1 or more that match our reference structure. And this, again, can be done automatically. So I'm just going to show what this looks like on our real data. So in each of the cases, I'm showing here the 5 structures that came out of our final heterogeneous refinement through all the steps of cleaning that I just mentioned. In each case, the first number shows the number of particles after junk detection that were available to classify. And the second number is the number that are left over after this cleaning procedure. And you can again see there's a huge variation across the data sets of how many particles are retained and how many are thrown away as junk in this process. It's also clear that in some data sets, multiple structures in the final heterogeneous refinement actually match our reference. And this is common if you're processing this kind of data. You may get 1 or 2 or 3 different structures that actually have the same overall shape, and the particles have simply separated into multiple classes, whereas in some cases it's just 1. Okay. So now we finally have hopefully a good clean set of particles that we can take on to the next stages of processing. Actually, I'm just going to mention again as another new feature in CryoSPARC, a tool and some tools that were necessary for the previous steps that I mentioned. And these are all released in CryoSPARC version 4.5. In CryoSPARC version 4.5, we have a new system and a new software sort of concepts called a volumes group. Volumes groups are super handy. They allow you to connect multiple and an arbitrary number of volumes from one job to another in a single connection. So again, for those of you who've used CryoSPARC before, prior to version 4.5, if you had, let's say, 10 classes coming out of a heterogeneous refinement and you wanted to connect them to another job, you would have to make 10 different connections. Now you can connect an arbitrary number of volumes from one job to another job downstream. And this is necessary for all of the automatic alignment and selection jobs that we have. And there are 2 in particular that I want to highlight that I haven't mentioned so far. One is Align 3D, and the other is Regroup 3D. And these are utility jobs that allow you to take a series of volumes, align them all to a reference structure so that their poses are all in register as well as hand flips. And finally, Regroup 3D that allows you to take a set of classes, let's say from 3D classification, and cluster those, the 3D structures themselves, based on their similarity to each other. So handy tools if you're working on heterogeneous data. Okay, so now with our particle curation done, we can move on to refinements. And here we can simply use non-uniform refinement in CryoSPARC, which is well known to be especially effective for smaller membrane proteins. In CryoSPARC, in the latest versions of CryoSPARC, our refinement jobs also produce a new set of orientation diagnostics. And in fact, there's also a separate orientation diagnostics job that gives a wider set of diagnostics that are all useful in assessing preferred orientation. I'll talk about those in a second. And here is where we can also, once we've done a refinement, we can use the Align 3D job that's new in CryoSPARC 4.5 to put our structures from ab initio and heterogeneous reconstruction in register with our original reference structure. And thereby we can use a mask, a single mask that again was created and used across all the different data sets, a generic mask that masks out the transmembrane region of our structure and allows us to do a local refinement, again, automatically, based on for each structure separately. Okay. So just a note about non-uniform refinement. In recent versions of CryoSPARC, version 4.4 and newer, we've had some substantial performance optimizations in non-uniform refinement itself that have led to around a 7 to 10x speed up in the specific part of non-uniform refinement that produces the adaptive regularization. And that's the part that actually takes our raw 3D reconstructions and adaptively filters them to optimally remove noise from different parts of the structure that have different structural characteristics. This speed up translates into, in many cases, often a 2x speed up in overall job runtime, and that can be substantial for many people. Of course, the effect depends on box size. Just an important note. And the orientation diagnostics that I mentioned, we have a set of new orientation diagnostics. Many of them are based on conical FSCs, which were introduced in tools like 3D FSC. We've defined a new metric that's called CFAR. It stands for Conical FSC Area Ratio. And we produce this metric now with every CryoSPARC refinement, along with a series of plots like the ones you see on the right, that helps to understand how good or bad your structure is from a preferred orientation perspective. The CFAR score is quite sensitive in the sense that it changes very drastically between structures that are good and structures that are bad. A CFAR score of 0.5 is what we recommend as sort of a threshold where below 0.5, you should be concerned that you have a preferred orientation issue that's affecting your reconstruction. Above 0.5, typically a structure is not affected strongly by preferred orientation. This measure, the CFAR score, importantly, is a measure of anisotropy that's sensitive both to the actual orientation distribution of where the particles are aligned, but also the distribution of signal. And this is a key distinction because it's common in imperfect data sets where you do have some junk particles and so on, that many junk particles are aggregated in certain orientation directions. And so it looks like you have signal, it looks like you have particle observations in those directions, but in fact, those observations don't contain useful signal. And so that distinction is captured in this score. So also, just as a note, in the latest versions of CryoSPARC version 4.5, we also produce a direct visual that shows you which views of a structure are rare in your dataset, and that can be used for targeting repicking based if you do have preferred orientation. Okay, so finally, before I show the results of actually processing our 8 datasets through this workflow, I just want to recap that the automatic workflow that we've talked about here doesn't include any manual steps. It can be started with a single click in CryoSPARC using the workflow software system that I mentioned earlier. And across all the datasets, we're going to test it out using identical inputs. So fixed job parameters, for example, particle diameters and so on. A generic reference that's used fixed across all the datasets, and fixed decoy volumes, as well as the fixed mask for local refinement. So you can imagine setting something like this up if you're working on a project that requires multiple structure determinations of the same or similar targets. You can set up a system like this based on the tools that we are talking about today. Okay, so now we'll talk about the actual results of processing this data. So just as a recap, here again are the EMDB deposited maps that are visualized here, as well as the resolutions with the deposited structures, or rather of the deposited structures. And here are structures that were automatically processed through the workflow that I just described in CryoSPARC from end to end without any manual intervention. I'll just flip through those again. Deposited structures, automatically processed structures. So we have the resolutions for each case written below. What you can see if you look across the 8 of them is that actually in 4 cases, we were able to get a nominally improved resolution of the overall structure. And I should mention that resolution estimates here are done using the same mask, the deposited mask for each case as the original authors used. So in each case, we can see that there are some data sets where we actually do better using the automated processing and some where our overall resolution is a bit worse. And of course, resolution is only one metric that doesn't tell the whole story, and often map quality in certain regions or particular regions that we care about may not track exactly with the overall global resolution. Still, it's notable that in the cases where we don't do so well, the difference in resolution is not great and the map quality is still relatively quite good. And in the cases where we do better, there are certain regions of the structures where the map quality is clearly improved. But this may not, as I mentioned, target the region of the structure that we're actually interested in. We can also look at our CFAR scores. So here are the CFAR scores in black, the CFAR scores of the deposited half maps, and in purple, the CFAR scores of our automatically processed half maps. So you can see in all cases, we actually either match or improve the orientation isotropy of our reconstructions, which is great. And finally, we can look at what happens when we do some local refinements. So here I'm showing just the transmembrane domain as a zoom in each case. And this density that you're seeing is the automatic processing result using non-uniform refinement. And next, I'll show the automatic local refinement. So I'll just flip back between those again, non-uniform refinement and local refinement. So you can see that in many cases, the difference is quite stark. Local refinement dramatically improves the density quality. For example, in this case, 11,350 between the non-uniform refinement structure and the locally refined density, quite improved. And in many cases, the ligand structure is also improved. And I'll just quickly show the ligands from these different structures. And these densities are from the automatically processed local refinements, the atomic models coming from the PDB structures that were deposited. So clearly, in this case, our automatic processing workflow is getting us to the point where our density quality is actually really good and interpretable for use in a larger loop, for example, in drug discovery, where perhaps these are different ligands or different structures that we're testing or looking at. And we can now start to imagine a world where the structure determination is actually automatic, and it now remains to analyze these results and determine the biological implications of the binding pockets and structures of these ligands, for example. I also just want to very quickly touch on run times. These are processing times for each of the 8 datasets. Each dataset is processed using only 2 GPUs in CryoSPARC for preprocessing, and only a single GPU for all of the reconstruction tasks. So that means each one of these datasets can be processed with just two GPUs at a time. And of course, the pre-processing actually takes up quite a large fraction in many cases out of the total runtime. And that can be parallelized really trivially with more GPUs available, or that pre-processing can be done in real time. So you can see that in many of these cases, the automatic processing can be done in less than a day, and there may be further optimizations available as well. And of course, the larger data sets do take quite a bit longer. Okay, and finally, we actually just in the last few days managed to get a few more results from testing this workflow. This is all very fresh. So we actually have results on 6 more data sets, and I don't have figures for all of these. So what you're seeing here are just the deposited EMDB structures. And in black, again, the deposited resolution for each of these 6 additional test cases that we tried. Again, using the exact same parameters, same inputs, same reference structures, and so on. And next are our automatic processing results in terms of resolution. So here again, you can see that in one case, we were substantially above the deposited resolution, in some cases a fair bit below, and in some cases matching the deposited structure. But again, these are data sets that which we hadn't seen in the development of this workflow, but were good test cases to see whether our selections of references and parameters would generalize to a new set of structures. Oh, and the CFAR scores as well, similar story that in most cases we're able to improve the isotropy of our reconstructions. So finally, just to conclude, after showing all of this, it turns out, and we think this may be important for the future, that automated processing can provide interpretable high-quality density maps, even for a challenging class of targets like GPCRs. With the tools that we've shown, it becomes simple to set up a workflow for other classes of targets or repeat target structure determination. If you're working on a specific GPCR or a specific ion channel, but you need to solve 10 structures and so on, it becomes simple to set something like that up where the processing can be automatic. And of course, there are possible further gains in resolution and map quality, likely, for example, with things like local classification and perhaps other advanced or manual processing for each dataset. Awesome. And with that, I will jump forward. I actually had a few slides in between, but I'll just jump forward to thank you slide. Oh, a few more slides. Thank you. Yes. And happy to take any question.
Hi, thank you, Ali. That was a very interesting talk and definitely very impressive resolutions as well. I was just wondering if, like, using the micrograph denoiser, if that's been compared to other particle picking softwares and how the resolution has improved while using that as compared to using something else.
For sure, yeah. So I can comment on that a little bit. We actually, during the development of these workflows, tested out a number of different particle picking strategies. And before we had finished developing our junk detector and denoising techniques, we initially tested with using Topaz for particle picking. So in an automated case, what we were doing there was producing particle picks using blob picking and then selecting good classes from those with the automatic 2D selection, and then using those selected particles as training particles for Topaz particle picking. And we found that that did work across the different cases, but the resolutions and results were actually worse than using our denoising and the workflow that I showed here. We didn't dig too far into it, so that's not a blanket statement to say that other particle picking techniques can't work. And it may be possible, for example, to train, if you're working on GPCR specifically, to train a GPCR specific particle picker that might work across the class. We haven't tried that yet, but that could be interesting for the future as well.
Okay, thank you. I think we have a couple more questions. So I'm going to try and go over them in order. I think Jinghao was first, and he asked, after a new refinement, how to classify more to get only ligand-bound particles, or how to classify ligand states in heterogeneous single particle cryo-EM datasets. Does 3D classify without alignment work?
Great question. Yeah, we're actually working on a new set of experiments on top of these results where we're trying to see if it's possible to perform local classification of the transmembrane region and the receptor binding domain specifically, especially for the purpose of improving ligand density. I don't have results for that I can show yet, but we do know that on a few data sets, at least, the ligand density is substantially improved by doing local classification. And for that, we do use the 3D classification without alignments in CryoSPARC. So that's the 3D classification job. The parameters of that job that are important, or rather some parameters of that job can be quite important when you're doing smaller local focused classifications. But I unfortunately don't have any results to show for that right now.
All right, thank you. And then Mohammed is asking, next step of ref-based 2D selection is template picking. And is there any way to choose more parameters for ref-based select 2D classes like ECA resolution?
Sure, yeah.
And is there any way to export and import workflows?
Sorry, I didn't catch the last part. Any way to import or export?
The workflows.
The workflows, yes. So on the first part, reference-based autoselect 2D that I showed, that we actually used here in this data is an upgraded, improved version of what's been deployed in the public CryoSPARC. So there will be a new version of reference-based autoselect 2D that we think works quite a bit better. It doesn't rely on the other metrics like ECA and the other metrics that we get out of 2D classification. We find those to not actually be quite reliable in selecting 2D classes, but it uses an improved matching score that actually works a fair bit better than what we've released so far. So that'll be coming soon. And on the second question, yes, workflows are fully exportable. So they're very simple to export. I'll just jump back to the workflows page. You can export a workflow in a JSON format. So you just get a single file that contains your workflow. You can send it to someone else and they can import it into their CryoSPARC instance and have your entire workflow available for them to use. You can also put those on GitHub or share them in any other which way that you like, and other people can make use of those. And those exported workflows will also contain your annotation. So if you added a description or you had default parameters or flagged parameters or locked parameters that you set in your workflow, those will also be exported as part of the file.
All right, thank you. I think Matt had a question as well.
Yeah.
Hello. Hey, Ali. How you doing, mate? You good?
Good. How are you? Good to see you.
Man. Yeah, love your work. We've obviously been using CryoSPARC since 0.6, I think. I started using it. Anyway, it's good stuff, mate. Hey, but I've just got my question is around your workflow that you showed in your these data sets, and it's really cool. But are you worried that you're going to lose too many particles during your ab initio classification step? The only reason I ask this is that I've noted that when we do sort of multiple class ab initio, there's still a lot of good particles that get clustered into some of the other, let's call them junk classes or poor ab initio classes. And so what we would often do is go back and do another 2D classification step on some of the junk, the junk classes.
I see.
But I guess my question, and maybe it's a room for improvement potentially, but why not in this particular type of workflow, would it be possible to implement sort of a hard classification step at the end of the ab initio, such that you don't lose actually potentially good particles that are getting filtered out into, or getting sort of driven towards poorer classes? Is this something that you guys are thinking about, or have you noted this behavior yourself?
That's super interesting. I don't think that we have found that ourselves, but that could be because right now our testing of this kind of a workflow is limited to public Empire datasets, and the ones that are deposited to Empire still, even though they're challenging, tend to be the sort of cleaner and better of the GPCR datasets, I'm sure, that are being collected in the wild. So it could be that we haven't seen that ourselves yet, but that's a really important point. I think one potential thing that could be interesting to think about there is that all the particles that are classified during ab initio reconstruction, they are, as you mentioned, they're not hard classified. They're soft classified until the very end. And we only put them into classes based on whichever structure out of the different ab initio classes that there are that they have the highest probability for. But a particular particle may have, let's say, 20% probability of belonging to the correct structure and 30% to a junk class. And it could be that we should not actually throw away those ambiguous particles until a later stage. And maybe those are the particles that you're mentioning that are still good particles, but falling into the wrong classes during ab initio reconstruction. Unfortunately, that's not something that we've investigated in a whole lot of depth where I could directly say whether that class probability is actually a useful way to find those particles. But it's really great to know, and I think we'll definitely take note of that and take it back to our team for more experiments, is how much or whether or not we're losing good particles in this kind of a scheme. And we definitely happy to get any kind of feedback on this at any time for sure.
Anyway, thanks a lot, mate. Cheers.
Definitely. Thank you.
Oh, so Meenakshi?
Hi, it was an awesome talk, beautiful talk. I love using CryoSPARC.
Great.
Just a quick question. Could you please say something on the symmetrical refinement thing? Like if you have, for example, a C2 symmetry in your data set, how would you refine that for a better resolution?
Sure. So I can talk about that just very generically. Generally, if you definitely know biochemically or from some other means that you absolutely have a symmetric object, a symmetric particle, you can go ahead during the refinement job and apply that symmetry. There's a parameter called symmetry, and you would just type C2. CryoSPARC will attempt to automatically figure out the symmetry axes in case your volume is not aligned in the box to the symmetry axes. It'll try to figure that out automatically. There's also a separate symmetry alignment utility in case that doesn't work. And then the symmetry will simply be enforced during the refinement, which will mean that every particle is used twice. So in a C2 symmetric world, every particle would be used for two different projections of the structure. That's great if you know that you have symmetry, but in many cases, symmetry can be sort of an elusive concept where maybe there's actually a pseudo-symmetry. The particle looks symmetric, but there's part of it that's not quite symmetric, or you have heterogeneity that, let's say you have two domains that make up a C2 symmetric object, and each one has some heterogeneity. If one of them can flex without the other one flexing, then your heterogeneity will actually break the symmetry. So in those cases, there are other tools that were recently released in CryoSPARC and have been used in the cryo-EM field for many years, generally known as symmetry relaxation or symmetry expansion, which can help in those cases where you have a pseudo-symmetry or you're more interested in a subunit's flexibility or conformations or something like that, that break the symmetry of the overall object. So hopefully that answers the question somewhat.
Yes, definitely. Can I ask one more question if
Sure,
there's time? Have you tried like processing inactive states rather than active states?
Yes, unfortunately I don't have those results in the slides. We do have some very early results in processing the inactive states as well. It's definitely more challenging, but the automated workflows using the same pattern that I showed here do work. We're still collecting more data there, aggregating more data, I should say, and trying to see how far we can go.
Thank you.
No problem.
So I think we have time for one more question. Sophia?
Hi. Thanks for the super interesting talk. My question ties in to Meenakshi's question about inactive or maybe G protein-less states of these GPCRs, and also ties in with Alyssa's question in the chat, specifically about these decoys that you might be using for inactive states. Are they similar? Are you generating them in similar ways to the active state? And to tie in Alyssa's question, if you're generating them from that data set itself or outside of that, are the results varied?
Yeah, that's a great question. So unfortunately, I don't have results to speak to those in a precision, but what I can say is on the inactive states, we do use the same concept of decoy classes, but the decoy classes are created specifically for the inactive version
Yep.
or the workflow. So we would have an intact inactive structure that is ready, and that's the overall object. And there, of course, I mean, different inactive structures will have different extracellular domains and stuff like that, but
Sure.
those are all averaged out into an overall coarse correct structure. I know that we do produce decoy classes in a similar way. So for example, erasing everything except the micelle, and then erasing the micelle and leaving everything else. Like, I think in the inactive case, if I'm not mistaken, we leave the transmembrane region, but just erase the micelle. And we do also generate these random noise classes. The way the random noise classes are generated, by the way, is very simple. And you can do this on any data set. You just start off an ab initio reconstruction, let it run for 100 iterations and kill it. And those structures that are generated at that point are extremely noisy and totally nonsense, but they're at least at the same grayscale level and overall like size of object. And so it can be a very useful way to generate decoy classes just automatically in CryoSPARC.
Yeah, great.
We do, once we're more settled on how this all works, we do plan to sort of operationalize this a little bit more simply so that people can try an approach like this more easily. By the way, for any questions like this and anything else that ever comes up about how to do processing or any tips or tricks, you can always contact us and reach us through the CryoSPARC discussion forum, which is a great resource for anyone using CryoSPARC. There's tons of activity, many users, advanced users, new users who comment there and share results of their processing and their stories. And our team is also actively monitoring the forum on a daily basis.
Amazing. Thank you so much.
Thank you.
Thank you so much. So I know we have a couple more questions from the audience, but I'm trying to stay conscientious of the time. I think do you have time to stay for a couple more questions, Ali, or?
Yeah, I'm good to stay for a couple more, yeah.
All right, so I'll just read. So I think we've covered Alyssa's question more or less. So someone asks, how do you remove micelle or nanodisk density?
Great question. So I'm guessing this is in reference to the generation of these decoy classes, in which case that's done manually using, for example, the volume eraser in Chimera, which is a handy way to do stuff like this. You can also use the segmentation tools. It may be possible, depending on your structure, to use thresholds and dilation and padding using the volume tools jobs in CryoSPARC to get rid of the micelle, but that's usually less robust than just doing it manually if you only have to do it once. Hopefully that answers the question.
And one more from Mohammed. So is there any way to deal with low CFAR but high SCF value? What are the best strategies for dealing with those issues?
Great. That's a great question. Unfortunately, I don't have the slides in this talk for more deeper results, but we are planning on making those public soon for how the CFAR score works and how you can actually use the outputs of orientation diagnostics to identify the rare views that you can then go back and try to specifically pick for using, for example, denoise micrographs, where it becomes much easier to identify specific views. So we do have results, for example, on this hemagglutinin trimer dataset, where even in the very preferred orientation case of the untilted dataset, we can actually take the views that are missing from our new orientation diagnostics tools, go back and repick those specific views and particles from the dataset and improve the CFAR score substantially by doing so. Generally speaking, the SCF score is not highly sensitive to the same sorts of pathologies that the CFAR score is sensitive to. So it's quite common to have a low CFAR score, but a high SCF score. I don't know if there's any specific advice I can give in that case other than going back to particle picking, or if the most unfortunate case, going all the way back to sample preparation.
Okay, thank you, Ali.
Thank you.
So I think this is all for today. So on behalf of everyone, organizers, audience, and myself, thank you so much for joining us here today and sharing your amazing work. And thank you everyone for joining us for this seminar today, and we will see you at the next one. So yeah, thank you.
Thank you all so much.
Bye.